And we're live. Are we? Yes, we are live. We're live. Laura, thank you so much for being here this morning.
Thank you so much for having me.
Could you kind of talk about the premise of your company and kind of to talk about My Personal Therapeutics and talk about the premise of the company and how you come up with the idea of building what you're building.
Sure. Well, good morning, everyone. I'm Laura Towart. I'm founder and CEO of My Personal Therapeutics. We're a London-based company that's developing novel personalized drug combinations for each cancer patient. And so the technology was developed at Mount Sinai Medical Center in New York with over a decade of clinical and basic research. And the technology was invented by Dr. Kagan, and who was at Mount Sinai, was the leader of the Center for Personalized Cancer Therapeutics. Now he's actually taken a position at the University of Glasgow. But the technology is really based on the idea that tumors are very complex. They're driven by multiple mutations, not just one single cancer mutation. And so the current standard of care is really, and kind of the best standard of care is targeted chemotherapy, where a patient would have either a mutational panel done, like a Foundation 1 test, and would identify a leading cancer driver like KRAS, and then a therapy would be directed to target KRAS specifically. And that was a major advance in cancer therapy. But one of the issues is that cancers are much more complex than that. This is something that we know now, that multiple cancer drivers are driving tumorigenesis. So the problem is when you're hitting one mutation, all of the other mutations start to strengthen and grow around it, which brings us to tumor resistance. It makes the patient stop responding very well to that chemotherapy. So this technology was really developed to address that issue and enable us to create personalized avatars for patients that incorporate many mutations, and then find a therapy that's directed specifically to that.
And the therapy, from my understanding, is PDP, right?
So the technology is called PDP. It's the personal discovery process. And I could explain that. The personal discovery process begins with the patient going to their oncologist at their point of care and getting a tumor biopsy, which would be sent to one of our partners for sequencing, for whole exome sequencing. So we look at not just the known oncogenes, but really all of the mutations that are driving tumorigenesis. And then that's where the personal discovery process starts. We do a very deep reading of the tumor genome, and we identify what are the drivers of that patient's specific cancer. We can select up to 20 mutations, and we engineer them into fruit flies, creating avatars. So for each patient would have a fruit fly that grows a tumor that's genetically similar to themselves. And this enables us to study the fly and try to understand which treatment combinations are effective against the fly's tumor, which would then also be effective against the patient's. So we use the fly avatars. We grow up about 500,000 avatars for each patient. And then using robotics, we do high throughput drug screening using all FDA-approved drugs or EMA-approved drugs alone and in combination. So we identify novel combinations that are effective against that patient's specific cancer.
I mean, so you caught me off guard a bit. Fruit flies. Fruit flies. So you guys are creating a model around fruit flies.
That's
how fast does the tumor develop within the fruit flies, and are there limitations to it, just given how small it is? And if somebody, I mean, apologies, I mean, I I don't quite understand this as in depth, but somebody from the outside, what if somebody said, are you better off not testing on mice or on another animal that has a hormone, that has a hormone or something? Could you explain that a little bit more? How does the tumor develop in the fruit fly?
Sure. So let me start with comparing it to mice, which is a very good point. So historically, and even today, mouse avatars are very important for understanding, tumor genesis and other diseases. The problem with and limitation with mice is you could only engineer 1 or 2 mutations in a mouse at a time. You can't incorporate up to 20 mutations, so you can't really model the true complexity. You could still learn some really interesting things, but you can't create, you can't have the mouse grow an avatar that's genetically similar to the patient. The other limitation is that we screen up to 500,000 fruit flies. There's no way you can screen 500,000 mice.
Whoa, okay.
You need like a military barracks to store them all, and then also the mice have a much longer life cycle. So
Okay.
it would take, you would be waiting years for the mice to develop the tumors and then do the treatment. So flies enable us to, they're a great tool for engineering mutations. We can engineer up to 20. Then we can grow so many of them, really many as we'd like, quickly.
Yeah.
They have a very short life cycle, so they grow the tumors within days. And we can easily make a tumor that's lethal. And the screen that we do is a lethality screen. So all of the flies will die from the tumor, except the flies that are getting the right drug combination.
Interesting. And so in a vivo environment, a fruit fly is somewhat similar enough to a human to model your to model your technology or your drugs on.
Yes. So, I mean, there are limitations. I mean, the fly in a human is a human, but we benefited from over a century of genetic studies on fruit flies. Really, all of the oncogenic processes were first identified and named in fruit flies, all of the main oncogenes. So there's enough similarity that enables us to create as close to a human-like tumor in an in vivo model. There's no other model that you can have a more representative tumor in a living organism.
That's really interesting. And to what extent can you test? I mean, so what tumors are really better modeled in flies? And at what point does fruit flies become a not so good model to model certain cancers? I mean, could you clear that up for the audience?
Of course. So one of the most conserved kind of organ systems from flies to humans is the gut. And so anything from the salivary glands, esophagus, GI system to the colorectum, or in the UK you call bowel cancer. So that's very conserved. So we really focus on gut-related cancers first, because that's where the model was really mainly developed at Mount Sinai for, and we have the most clinical data to support that the effectiveness of treatments developed using the fly in patients. We are also able to provide treatment recommendations for patients with lung cancer and many other cancers. But because we're a digital health company, we're building digital health tools. We're really trying to focus on a few groups of cancers first, and then we'll expand to other tumor types.
And what cancers are they?
So any gut-related cancer, like I mentioned, lung cancer, those are the two major groups, and then rare genetic cancers. And many patients with rare cancers find themselves without any treatment options, and we can provide novel treatment recommendations for them. And so we find a lot of patients coming to us with rare cancers. I'm happy to help them.
That's interesting. In terms of the cancers, I mean, so do you guys implant the tumors into the fruit flies? I mean, could you kind of clear up how that works, and also clear up what the process of building out a model kind of looks like, from when the customer comes to you to the point where you can deploy or prescribe certain drugs?
Sure. So the first question. So we don't implant the tumor in the fly. So what we do is when we identify which mutations we want to incorporate in an avatar, we create a construct. So basically, it's a vector with a bacterial backbone, and it's micro-injected into the embryo of the fly, and then the fly will grow up, and we can direct the construct to the region of the fly corresponding to the region where the patient has the cancer. So if the patient has bowel cancer, we can direct the construct to grow in that area of the fly, the hindgut. And so it's using the tools of molecular biology more than kind of a tissue implantation. And then the process for a patient would approach us. They can pay directly on our website after we speak with their oncologist and just understand that the oncologist is on board. The patient is a good candidate for this. And ultimately, the oncologist will be treating the patient. So the oncologist needs to, feel comfortable with the idea that we're creating novel combinations. And so we work with the patient, and we work with the patient's oncologist as well. So the patient then can download our app, which helps educate the patient throughout the process as to what the heck we're doing and how we're creating their avatars, just to provide the patient a little bit of comfort as well, to let them know for each patient, we have a large team of scientists that spend a lot of time focused on trying to find the best combination for them. So it's a very in-depth process with scientists of many disciplines that are working to really develop the best treatment for them. So we want to show that to the patient. We want the patient to feel comfortable that they've made a great decision to really be pioneers in kind of innovations in cancer therapy. So then the patient would have a biopsy with their oncologist, send it to our sequencing partner. Then we do the analysis, and then we select the mutations to engineer into the fruit fly. We expand the fruit fly line so that we have about 500,000. And then we do drug screening using the robotics. And then the patient would get a treatment recommendation report. One thing I didn't say, the patient, after sequencing, we do provide the patient and oncologist with an early report. So this report would just be based upon their whole exome analysis. And so some of the information that we learned from the whole exome could help direct the patient's care early on. It wouldn't be completely personalized, but it would be kind of on par with what other personalized cancer companies offer today, maybe a little bit more in depth. But then we create the avatars, and then the later report is a true personalized drug combination.
Wow. And would you be able to shine a light on how many drug combinations you guys are experimenting with?
Sure. So we screened the whole FDA or EMA approved drug library. It's about,
wow.
Yeah, it varies every day, and it depends on which specific groups of drugs you include, but it goes up to about 2,000 drugs. So we screen each drug alone and then in combination. So it's tens of thousands of drug combinations.
Wow. Why is the fruit fly a better insect or living being to test these to develop a model around around colorectal cancer, or the cancers that you guys are kind of specializing in?
Well, so as I mentioned, compared to mice, they're small and genetically malleable. And they also, because we give the flies the drugs in the food, flies, imagine they're eating very little amounts of food compared to other animals. So the drugs, some of the drugs are very expensive, and we just have very tiny quantities, which is great. There are other models. So there's zebrafish is another great model and good tool for understanding cancer. But zebrafish are also they're quite large fish. In order to create zebrafish avatars, you need major aquaculture facility, and so also you have the same kind of limitations where you can't do high throughput drug screening.
That's really interesting. I mean, how long does it take to kind of build a model for a tumor type you haven't done before, and also for could you provide a comparison between a tumor type you haven't done before and then also a tumor type you guys can really help with, like colorectal cancer?
Sure. So the idea is that patients with similar tumor genomic backgrounds, so patients that have similar mutations that are driving their tumor, will respond to the same drug combinations. And this is something that we've understood from the data that was gained at Mount Sinai. And so the idea is that after we've done a certain number of patients, an incoming patient, we can say, oh, this patient's tumor profile is just like patient number 2. And patient number 2 had this drug combination. So we could recommend that right away for the incoming patient. So if an incoming patient is lucky, we'll have a patient like them already in our database, and we can provide a much quicker treatment recommendation. So with GI cancers, we're starting to generate a very large data set. So it's likely that if a patient comes in, we already have enough prior data that can help us. So it could take somewhere around 3 to 4 months to develop a treatment recommendation for a patient with GI cancer. For a patient that has a tumor type that we haven't had a lot of experience yet, it could take longer. And so it's really, it's something that we're working to bring down the time. And there's a lot of different ways in which we can do this. The first step is the patient's getting sequencing, which is done by a third party. So we're trying to identify the best third-party sequencing facilities around the world that can give fast turnaround with great results. One of the other things we're building, which is kind of cool, is so you could imagine that when we have 500,000 flies, each with the patient's tumor, engineered inside, and then we're doing tens of thousands of drug combinations on each. Someone at the end of the day has to count each fly vial and say, okay, how many flies with this drug treatment are alive and how many are dead? And that's how we understand if that drug combination treatment worked. And so right now that's done by a human vision. So if someone has to go and count each of the thousands of fly vials, and see how many flies of each type of drug. So it's a horrible job. No one wants to do it. And we're building image analysis software. And so this will, that's a major constraint, is having to have, as we get more patients, imagine having so many patients in the pipeline, so many vials around with flies in them that need counting. So it does become a potential source of backlog. This is going to enable us to not only enhance the speed, but also enhance the quality. So computer vision doesn't make as many mistakes, as it's not prone to human error. So we're trying to really take a lot of it out of a lot of potential sources of error or kind of time delay out of the process. One of the other things is right now we're just counting, whether the fly is dead or alive, but some of the tools that we can build into the image analysis are really looking at the overall health of the fly. So instead of just being dead or alive, we can have measures of, is the fly healthy longer? And one of the ways you can tell that is if the fly is fat. So if the fly is bigger, it's a happier fly and healthier fly. And it'll enable us to make distinctions between the drug combinations.
So you guys are basically, how do you know what drugs to look at? So what happens when your algorithm spits out a recommendation for a drug combination that's approved for a different indication? I mean, do you anticipate in the future that your business model will revolve, an element of your business model will revolve around working with pharmaceutical companies and kind of providing them with an understanding of other use cases a specific drug can have?
Yes, that's exactly correct. So all of the drug combinations will be novel. And we know now that, one of the big schools of thought in oncology is that not looking at cancers by only tumor type, but grouping them by, leading oncogene. So like if there's a KRAS positive cancer that has a drug that is approved for, let's say, prostate cancer, but we find that a patient with this specific KRAS type mutation has colorectal cancer, benefits from this drug combination, this drug in combination with another drug, then of course we would communicate with pharma that there's a potential new indication for their drug, new opportunity for them in the marketplace.
This is interesting. I mean, does your company work with UK I mean, I'm assuming your company just works in the US, just given the fact that it's privatized, right? Or are you guys working with the NHS or private providers here in the UK?
So we work with a private oncologist all around the world.
Okay.
And so it is, like you're right to say, it is a little bit difficult in the UK because with the NHS, this is something that wouldn't be available today. But we received grant funding from Innovate UK to do a really amazing study with Professor George Hanna at the London In Vitro Diagnostic Centre, Hammersmith Hospital. And so we're going to be enrolling some GI cancer patients and creating avatars and doing screening in that setting, so that we can generate the data that will need to go to the NHS in the future to try to get this incorporated.
That's incredible. I mean, what are the use cases are there for fly avatars? I mean, what's the future of fly avatars? What are there collaborations you guys have been working on? I know you just mentioned Innovate UK. Yeah, could you talk about the future of the of fly avatars and what you guys are working on right now? What you have been over the last couple of years?
Sure. Well, it's very exciting. So the possibilities are kind of endless. One of the cool things that we could do, which was pioneered at Mount Sinai, is create diabetic flies that also have cancer. And we're hoping to do one of the largest and first studies to show that patients that have diabetes and cancer should receive completely different drug combinations than a patient without diabetes. And so that's something that we're going to be doing. So we can model comorbidities like diabetes. We could model if a patient is obese, we can make an obese fly. These things matter when looking at personalized medicine, and so that's something that's exciting. We could also create rare genetic disease models. So not just modeling rare cancer, but modeling rare genetic disease, which is something that we really have a strong interest in doing. Patients need drug combinations that have no other alternatives. We could also create flies that have different neurodegenerative diseases. So really, there's a lot of kind of advances in medicine that can occur to make personalized avatars.
That's incredible. And in terms of your company as a whole right now, what interesting things are you guys up to? Are you hiring? Are you fundraising?
Sure, both. So we are in the process of fundraising. Please definitely get in touch if you're interested in investing, and we are also hiring. So we are looking for kind of a unicorn person, so someone that has a background in machine learning, deep learning, statistics, with a kind of genomic background, some genomic data set background, and then even more perfect would be if the person also has experience dealing with oncogenic data. So we'll see. So we're definitely hiring that. I mean, if anyone's interested, finds what we're doing exciting, please do get in touch, and happy to have a conversation, because we will be hiring across different domains soon.
Yeah. You said you were fundraising. I mean, would you be able to shine a light? You don't have to if you don't want to, but shine a light on how much you're looking to raise and what you're hoping that the funding could facilitate within your company.
Sure. So we are revenue generating today. So we're currently selling to patients and oncologists, and we're also selling to pharma. So we're able to do some pharmacological screens. So we can take an investigational drug, add it to our screen, and provide really valuable information for pharma. So we're revenue generating, and we would like to raise the funds to be able to scale. So we want to be able to increase the capacity of our current laboratory by expanding, and potentially have one laboratory that serves patients and one laboratory that serves pharma, and really to scale up our team to enable growth. We're raising. Our target is around between 3 to 5 million. So we're kind of open to like a 3 million round, but a lot of investors now are interested in investing more, taking a bigger chunk of the company at once. So we're flexible, and really this fundraising round we see would bring us to break even.
Interesting. And in terms of commerciality, I mean, I don't quite understand the. I mean, how are you guys commercializing your technology? I mean, I find it hard to understand because obviously in the UK everything is most things go through the NHS. I appreciate there is a privatized sector, but I mean, what parts of the world are you guys working in right now, more specifically?
So really, we can work anywhere. We've just signed an agreement with a group and to bring our technology to South and Latin America, where we signed another agreement with a partner to bring the technology to Australia and Asia. We are working in the United States. We're working in the Middle East, where we're about to start a study with a major hospital in Saudi Arabia. Also, the Middle East is very kind of used to private and innovative cancer and other technologies services. So we're working kind of strongly in MENA and Europe. And in the UK, I mean, we are able to offer the technology through, through private oncologists. And I think there are no regulations that kind of govern our personal discovery process. So we're able to offer treatment recommendations through oncologists as a decision support tool.
And if people do want to have part of your treatment or have some of their cancer models modeled, can they reach out directly to you?
Yes. Yeah. So on our website, there's kind of tabs for a patient, a physician, and then there's kind of like a big how to order button. And so So if a patient would click that I'd like to order, I'd like to take the next step, they get in touch with us. We need to find out more information from their oncologist, and then we go through that process, which is very fast. They can fill out most of the forms that we need through the website, and then we get started right away.
Interesting. And so, final question. I mean, in an uber optimistic world, 24 months from now, what do you hope to have achieved?
Good question. So within the next year, hopefully, in the next 6 to 12 months, we hope to have our first predictive model for all GI cancers, and really then we can start doing some validation studies. And so in the next 2 years, we hope to have that big data product kind of validated and on the road to reimbursement in the United States, and then kind of also then shortly after having that tool for other cancers like lung.
Amazing. Is there anything else you want to kind of shout out before we wrap this up?
No thanks, but anyone who's listening, if you're interested in investing, or if you know someone that has cancer interested in learning if we could help them, please do get in touch.
Amazing. I really appreciate you jumping on this morning, Laura. Take care.
Thanks so much. Bye-bye.
Bye.